Every second, an invisible engine powers the global carbon cycle, turning sunlight and air into the physical fabric of life. This is the Calvin-Benson cycle, the biochemical architecture of global carbon fixation. While historically called the dark reactions, this system is completely reliant on and strictly regulated by light. Unraveling this pathway was a milestone in modern science. In 1950, a team at Berkeley Lab, Melvin Calvin, Andrew Benson, and James Basham, pioneered the use of carbon-14 radioactive tracers paired with 2D paper chromatography. By tracking metabolic intermediates in real time, they mapped out the complete pathway, earning Calvin the 1961 Nobel Prize in Chemistry. The entire operation functions strictly within the stroma. It operates as a continuous input/output loop, relying completely on the energy currency generated by light-dependent reactions. By consuming ATP and NADPH, this fluid-filled space serves as the physical factory floor, where atmospheric carbon dioxide is transformed into complex carbohydrates. Phase 1 acts as the system's intake valve. A 5-carbon sugar called RUBP acts as the primary carbon dioxide acceptor. The massive, slow enzyme Rubisco catalyzes this critical step, fixing a single molecule of CO2 to RUBP. This highly unstable 6-carbon intermediate instantly splits, yielding 2 stable molecules of 3-PGA. Phase 2 begins the structural reduction through a heavy energy investment. First, 3-PGA is phosphorylated by the enzyme phosphoglycerate kinase. This process requires the direct consumption of ATP to manufacture 1,3-bisphosphoglycerate. This evolutionary step directly mirrors the exact reverse reactions found in cellular gluconeogenesis. Powering the core of the cycle requires reducing power. Driven by the enzyme GAPDH, 1,3-bisphosphoglycerate is reduced into glyceraldehyde 3-phosphate, or GAP. This step utilizes NADPH as the critical electron donor. GAP is the primary high-energy intermediate used for all downstream synthesis of plant glucose and starch. Next is product allocation, governed by a strict yield ratio. For every 6 carbon dioxide molecules fixed, 12 GAP molecules are generated. However, only a harvest of 2 GAP molecules exits the cycle to serve as building blocks. These exported molecules form hexose precursors, ultimately assembling into glucose and starch. The remaining 10 GAP molecules carry a heavy recycling burden. They must be completely rearranged to regenerate 6 RUBP acceptor molecules. This requires an enzymatic assembly line featuring transketolase, aldolase, and SBPase. A final primer step uses PRK and an additional ATP investment to reset the system to its starting state. The true metabolic cost of this system is immense. To fix carbon and output the necessary building blocks, the cycle demands an input of 18 ATP and 12 NADPH molecules. Generating this energy requires the front-end light reactions to absorb approximately 48 photons of light, showing the true scale of the process. To prevent wasteful energy use at night, plants evolved an elegant biochemical light switch. In day mode, light-driven electron flow reduces thioredoxins, breaking rigid regulatory disulfide bonds to activate key enzymes. In night mode, these disulfide bonds reform, locking targets like Rubisco activase, PRK, and GAPDH into an inactive state. This system is reinforced by a supramolecular control mechanism called the CP12 molecular lock. In the dark, the unstructured CP12 linker acts as a physical tether, structurally binding GAPDH and PRK into an inactive complex. When the light returns and the NADPH ratio rises, the complex spontaneously dissociates, releasing the enzymes to resume the cycle. The engine has a massive flaw: the Rubisco dilemma. Rubisco's active site cannot perfectly distinguish carbon dioxide from oxygen. Fixing oxygen initiates a wasteful process called photorespiration, producing a toxic byproduct called 2-PG. Salvaging this lost carbon drains immense energy across the chloroplast, peroxisome, and mitochondria. Evolution has engineered a solution. While standard C3 plants suffer a passive CO2 diffusion mechanism and up to a 25% energy drain from photorespiration, C4 plants feature a mechanical upgrade. C4 plants actively concentrate carbon dioxide directly around Rubisco, reducing photorespiratory losses to near zero through specialized metabolite pools. Today, agritech is upgrading this ancient engine. Scientists use targeted mutagenesis to improve Rubisco's catalytic rate and CO2 selectivity. They also overexpress regenerative enzymes like SBPase and PRK to increase biomass, and introduce synthetic genetic bypasses to shortcut photorespiration, aiming to boost crop yields by up to 40%. The final frontier is artificial photosynthesis, harnessing the Calvin cycle architecture to convert sunlight and CO2 directly into sustainable liquid fuels. The critical bottleneck remains the front end. Engineers must fully replicate the precise four-step oxidation cycle of the photosystem II complex, the Koch clock, to unlock synthetic carbon fixation.